Good day, this is Professor CBMD PhD, and today's Monday, June 29, 2026. It is 9:51 p.m. Eastern Standard Time, and today's research, H5N1 bird flu. So let's hop to it here. Scientists finally solved how H5N1 bird flu hid in dairy cows. Scientists cracked the mystery of bird flu's strange behavior in cattle, and the answer could help predict the virus's next move. Date June 23, 2026. Source: University of Pittsburgh. The scientific investigators and researchers uncovered why H5N1 bird flu attacks cows' udders instead of their lungs. The virus's preferred receptors are concentrated in mammary tissue. The breakthrough could help scientists predict future bird flu jumps and spot unusual infections before they spread widely. The outbreak first emerged in dairy cattle in the Texas Panhandle, where animals develop severe cases of necrotizing mastitis, a painful inflammatory disease that damages tissue in the mammary glands. Mastitis is a classical disease in milk production animals, and veterinarians were dutifully looking to all the usual suspects for the source, like bacterial pathogens. When the real culprit turned out to be bird flu, everyone in the field was caught completely by surprise. We hadn't even remotely considered that cattle could be a host for H5N1. Before the virus was identified, it spread from herd to herd, infecting cattle and contaminating their surroundings. If a cow is infected, it sheds a lot of virus into the milk. This raised concerns about occupational risks for farm workers. Also, there is a habit of feeding raw milk to domestic pets like cats, and there have been instances of cats dying which we studied previously. The researchers emphasize that pasteurization effectively destroys the virus, highlighting the importance of avoiding raw milk. The scientific researchers studied influenza viruses, focusing on how receptor biology influences which species and tissues can be infected. Influenza viruses attach to specific receptors on cells in a lock-and-key fashion. These receptors belong to a group of sugar-based molecules known as glycans. Earlier studies from other research groups suggested that flu-related glycan receptors were present in the noses, tracheas, and lungs of cattle, yet cows infected with H5N1 were not developing the respiratory disease researchers expected. That discrepancy suggested that a more detailed explanation was needed. Glycan biology is very complex. We realized that to understand what was really going on, we would need to use more innovative technologies and map out the fine detailed architecture that enabled the virus to bind the cells. The research team combined multiple techniques, including binding experiments, staining approaches, and ultra-high-resolution imaging to examine how H5N1 interacts with different tissues. Their analysis showed that not all glycan receptors function the same way when it comes to bird flu infection. The virus was able to bind only to a specific subtype known as N-linked sialic acid receptors. These receptors were found throughout the outer tissue of cattle, but were nearly absent in airway tissue. This made the mammary glands a perfect breeding ground for the virus. The discovery helps explain why H5N1 caused severe mastitis rather than respiratory illness in dairy cattle. The researchers believe their findings could do more than explain the cattle outbreak. The same approach may help scientists identify which animals and tissues are vulnerable to H5N1 before future outbreaks occur. We can preemptively screen different species and different tissues within them for susceptibility. For example, would they exhibit respiratory symptoms? Would they show only mastitis, as in cows? Or would they show neurological disease, as our team has shown in cats? The lesions learned could potentially help prevent us from being caught by surprise again. And this video was created for educational purposes only. Hope you enjoyed listening. My codename is Will. I'm Professor Christopher Brunet, leader of research scientist ufologist, psychic computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover. My passion is world peace. Have a great day. Good day, this is Professor CBMD PhD, and today's Monday, June 29, 2026.